Thank you everyone for joining today. It's great to be able to share some recent advancements that we've had at IDT in the last year or so. My objective with this presentation is really to start with a brief introduction on CRISPR, but I think most of the audience is already quite familiar with that. And then we're going to go through a whole series of new developments that we've been working on. One of our primary objectives in the CRISPR space at IDT is to identify common stumbling blocks that we see in our customers and help them solve those either with new methods and recommendations or with entirely new products and tools that can help circumvent some of those common issues we see. So just for those of you who are maybe a little less familiar with us, I always like to give a little bit of our origin story so you understand why we do things the way we do. As a company, IDT was founded a little over 30 years ago by Dr. Joseph Walder in Coralville, Iowa. And so Dr. Walder's primary areas of research centered around functional genomics, so siRNAs and antisense oligos. And IDT was really founded to help him generate those oligos for his own research. And so if you're familiar with those technologies, they really require synthetic DNA and RNA molecules that tend to have a lot of unique chemical modifications on them. Fast forward to today, the company's obviously grown substantially. We employ over 1,500 people around the world, and we're supplying well over 100,000 active customers. So those are people who have placed at least one order with us in the last year. Many of you know us for our primers. That's sort of our core technology, but of course, that's foundational to a lot of the things we'll discuss today. And that's sort of the foundational building block of why we have such an extensive R&D department. As you can see in the figure here, RNA and DNA oligos are what we've been doing for decades. But at the end of the day, that is how we make the rest of our products, right? So everything that IDT is involved in is based on that core competency around oligos. And our research team's job is largely to take that amazing technology of synthesizing nucleic acids and adapt it to whatever applications people want to be doing in the lab. So we have a few examples here. Obviously, we'll be focusing on CRISPR systems. The data that I'm sharing today and the product developments and all of that largely originate from our CRISPR development group, as well as our bioinformatics team. But we have a pretty extensive research team covering all of our other product areas as well. So that out of the way, I just want to quickly run through the basics of CRISPR so we all have the terminology fresh in our minds, understand the basic technique. On the screen here, you can see a very high-level schematic of how genome editing might work with Cas9. The reagents that IDT is really involved in, and as a research scientist, you have the most control over, are going to be your nuclease, which we're showing here is just Cas9, but could be something like Cas12, as well as the guide RNA, which isn't really shown in this figure. When you deliver those two components into a cell, they're going to locate a region of the genomic DNA based on sequence complementarity to the guide RNA. And at that site, you introduce a double-stranded break in the genome. And so this is really the process you have the most control over in a CRISPR experiment. What comes after that is largely dependent on how the cell responds to that double-stranded break and what repair pathways it uses to correct its genome. So one possible outcome, and probably the most common outcome, is going to be a repair that relies on the non-homologous end joining pathway you see on the left of the slide here. When this is done, you basically get a ligation of those two blunt ends back together. Because the process can be error-prone, you typically will end up with a small indel at the cut site. When you do this in a reading frame of a gene, you knock out the gene, and that's how we would accomplish a knockout. So this pathway is fairly straightforward, but obviously doesn't allow you to control what the final mutant sequence looks like. If you want to do that, you need to include a donor template. We'll talk a little bit about these later in the presentation because it's an active area of research for us. But the basic premise is that this is some sort of exogenous DNA that encodes for a new genetic material, shown in green here, and that can be incorporated at the cut site using a repair pathway called homology-directed repair. And so this pathway is dependent on sequence homology between your donor template and the wild-type genome around the cut site, and that's shown in light blue here. This pathway is a little bit more challenging, doesn't occur as often in cellular DNA repair, and also the addition of a donor template adds another reagent you have to think about. But obviously, having explicit control over what the mutant genome sequence is is of interest to a lot of researchers. In terms of the guides and then the nucleases, IDT fully supports two main systems for editing. On the left, you have Cas9, which I think is what the vast majority of people are currently using. And it's the most well-characterized genome editing platform, if you will. But we also support Cas12a, which you're seeing on the right. There are a number of differences between these enzymes and the guide RNAs they use, but I think that the main one to really focus on for the sake of today's conversation is going to be the PAM site, right? So if you're going to edit using Cas9, you need to identify a sequence of NGG, which is directly adjacent to the protospacer of your guide RNA. So PAM is protospacer adjacent motif, which is a fairly intuitive name once you know what it stands for. But this is obviously a limitation, right? If you don't have this NGG sequence, you just can't cut at that site with Cas9. And so this is one of the main reasons people might consider Cas12a, because it uses an alternate PAM of TTTV, which is useful in more AT-rich regions of genomes. Like I say, there are other differences, but for the sake of brevity and for today's conversation, I think that's the main difference. And again, because the focus of today's talk is really about advances, I just want everyone to be aware, as you're reading through the literature and you're looking through maybe protocols on our website or elsewhere, you're going to find lots of recommendations on how to do this, how to get these reagents in cells. At the end of the day, what you're ultimately trying to do is getting your nuclease, perhaps Cas9, perhaps something else, along with your guide RNA into the cell, right? IDT's recommendation is to use recombinant protein and synthetic guides, but just be aware that all of these other methods are also valid ways of editing genomes, pros and cons to all of them, and we can discuss that in more detail at a later time. But most of them can also be adapted to some other methods. So if you're reading a paper that uses a cell line expressing Cas9 and in vitro transcribed guides, the ability to use recombinant protein and synthetic guides is also on the table. So, in the simplest forms, the protocol looks somewhat like this, right? You're going to take your Cas9 protein, mix it with your synthetic guide, and that's what you then transfect into your cells using whatever method is appropriate. So that was just a very brief introduction. My goal was to sort of set the stage and make sure everyone was up to speed on their terminology. I want to spend the bulk of this time, though, talking about the new advancements here. And so this figure kind of shows the full CRISPR product line that IDT supports today. Everything that you see in light blue here is something that we already have on the website. So this sort of starts at the very early design process with design tools to design your guides. We have a number of guide RNA formats and donor templates, as well as a number of tools for analyzing your edits on the back end of your experiment. Today's talk is going to focus on the orange highlighted items here. These are things that are either very recently launched on our website or are coming in the next few months towards the end of the year. So we'll talk a little bit about custom guides. I mentioned we fully support Cas9 and Cas12, but there are lots of other CRISPR nucleases and other enzymes out there that require RNAs to guide them. We'll talk a little bit about some new proteins that we're going to be launching soon, including a mutant Cas12a variant and some fluorescently labeled Cas9s. We've been working very hard on expanding our offerings for donor templates for people doing knock-in experiments, especially people who are trying to insert larger constructs. We've certainly struggled to make some of those larger constructs through our synthetic biology group or other areas. And so it's been an area of interest for us to find easier ways to accommodate those requests. Some new tools for improving HDR rates. So we'll talk about that near the end of the talk. And then a new analysis tool that we'll be launching next month to help quantify editing using amplicon sequencing. So let's start with guide RNAs. As I mentioned, IDT fully supports Cas9 and Cas12a, and what that really means, for you as a CRISPR user, is that these are products that are on our website. They've been really highly validated, and we have a really robust understanding of how to make them and what the best practices are. So I won't go through all the details because it's not really relevant, but for example, with Cas9, we have 3 different guide RNA formats. And if you come and talk to us about which one to use, we have lots of information about how you select your guide RNA, how you design it, and what format is best. Same goes for Cas12a. We only have one option for this because it's such a short guide RNA. But again, we have really good understanding of how to make these. And so these are what we would refer to as catalog products, right? They're on the website, anyone can just go and order them. The reality though is the CRISPR space includes lots of technologies beyond Cas9 and Cas12, and I'm sure many of you have encountered those reading through the literature or even just sort of the popular media and the news talking about using CRISPR for COVID-19 diagnostics, for example. Those types of applications use enzymes other than Cas9 and Cas12. And so for a long time, we've been able to make those guide RNAs when people emailed us and requested those things. But we've admittedly struggled to do that quickly. Science only moves as quickly as you can get your reagents into the lab. And so we wanted to make that process easier. And so a few weeks back, we launched our custom guide ordering tool. This is basically meant to accommodate any guide RNA that's not Cas9 or Cas12. So common ones that we see a lot of requests for would be Cas13, which is used in certain diagnostic applications, or long guide RNAs used for prime editing, which we just call PEG RNAs. And so these guide RNAs contain both a Cas9 guide as well as an RNA encoded donor template to be used with a special fusion protein. It's a unique way of getting small mutations introduced into a genome. But it requires long guide RNAs, up to 150 nucleotides. And so what this tool lets you do is enter the full-length sequence of your guide RNA up to 150 nucleotides. We can synthesize them at some common yields that you're probably already familiar with if you know our guide RNA products, as well as the common chemical modifications you would find on these. Generally, we can make these within a week or so. So a much faster way to get your guides if you're studying some novel CRISPR systems. And that's on the website already. We've also been working a lot on proteins. So we want to talk a little bit about some new developments there. I think if you've seen our webinars or our seminars in the past, you'll know that this has been an area of research for some time. In my introduction, I hope gave you a sense of how IDT has been really focused on oligosynthesis for decades, and that's really what drives our quality in guides. But if you do molecular biology in the lab, you know that your specificity comes from nucleic acids usually, but the catalytic component of any assay you run is going to be a protein, right? Maybe a polymerase or a nuclease or something. And so we've invested a lot of effort in developing protein engineering capabilities to help solve that side of the equation. And so in the past, we've talked about our high fidelity Cas9, right? That's a mutant version of a wild type Cas9 that reduces off-target editing. We've also in the past talked about a mutant version of Cas12a that we developed called Ultra. The objective here was to improve the on-target editing capacity of this protein because the wild type protein is a little inconsistent when it comes to that trait. And so these are both products we've had on the website for some time, but we're developing a whole range of new ones. So one, it's not highlighted in orange, is the LB version of Cas12a. So LB and AS indicate the bacterial species that these proteins are derived from. We originally launched our Cas12a products with the AS variants, but we get lots of requests for LB as well. And I think you see this cited in the literature quite regularly. So that protein is going to be available as well as the existing AS version. So that'll be online in the next month or two. But I have some data to share on these fluorescently labeled versions, which I think will be of interest to a lot of people. And so anyone in the audience who is familiar with protein engineering knows that slapping a fluorescent tag onto a protein is often a great way to just make that protein no longer work. And that's at the core of the reason why it's taken us so long to develop fluorescently labeled Cas9 and Cas12. If you're not very, very thoughtful about the linker structure you use between the nuclease and the fluorescent protein, you can really negatively impact the nuclease's ability to cut the genome. And so we spent a long time developing and testing different linkers to connect those two components together. And I think the data on this slide really shows that effort has paid off. So the top panel here is showing editing efficiency at a number of sites in the HPRT gene. The blue bar is the wild type Cas9 that we currently sell on the website. So this is not fluorescently labeled. And then the green and the pink bar indicate fluorescently labeled versions, either GFP or mCherry. And you can see that most of the time we're able to maintain very similar levels of activity with those fluorescently labeled versions. You'll find the odd situation, for example, the one I'm highlighting here, where we see a small reduction in on-target editing activity with the fluorescent labels. But for the most part, I would say this is worth it, right? Because if you have a fluorescent tag on your Cas9, you're going to be able to sort those cells and identify the ones that have been positively transfected. And so you can really compensate for that small drop in activity by enriching for those cells. And so in the case of Cas9, we have pretty comparable performance between unlabeled and labeled protein. In the case of Cas12a, the story is actually even more interesting, although we don't have a great explanation for this. We find that adding a fluorescent tag actually has improved the activity of this enzyme, which is not necessarily what you would expect. Typically adding an additional amino acid onto the core nuclease is going to impair the function, but that's not what we see. And that's reproducible across quite a number of sites here. I think the take-home message for people who want to use this for genome editing is that the labeled version is obviously still going to do a very good job, if not a better job than the unlabeled one. And I think really the main reason that you're going to want to use fluorescently labeled nucleases is going to be in situations where you're struggling to get high transfection efficiencies. Because remember, when you do a CRISPR experiment, your goal is to get the nuclease and its guide into the cell where it can go into the nucleus and edit the genome. But if you're working with a difficult cell line where you can perhaps only transfect 20% of cells, that can make your downstream experiments quite challenging. If only 20% receive the nuclease, the other 80% are never going to be edited. And that can really confound some of your downstream experiments. So obviously we would first encourage you to try and optimize your transfection, get those numbers as high as possible. But if you can't, we do know, and that's what this data here is showing, that if you use a fluorescently labeled protein and enrich for fluorescent cells, you can compensate for poor transfection efficiency and actually see a boost in average editing rates. And so each of these locations here is just a different gene that we're targeting. The green bar is unlabeled Cas9, so that gives you a sense of what the editing level is in the absence of a fluorescent tag. And then all of these other bars from gray through to dark blue represent different populations of cells transfected with fluorescent protein. And you can see that if you gate your flow cytometry for the highest or most fluorescent cells, the medium and high populations, you actually end up with higher editing rates than the unlabeled Cas9 or the unsorted fluorescent Cas9. And so this is really the main reason you would use a protein like this. And the same premise applies to Cas12a. So a great tool if you're working with difficult cell lines that are hard to transfect. For the sake of completeness, I would also mention we have offered fluorescently labeled guide RNAs for a very long time. And if you're familiar with our two-part guide RNA system that requires a cRNA and a tracer RNA, we've sold a version of this tracer RNA that has a fluorophore on the 5' end for a number of years now. That fluorophore is called ATO550. It's a red end dye. For this application, it works really, really well, but we certainly realize that there are times when that color is just not suitable based on your experiment. People have been asking for new colors for a long time. And so we'll be adding additional labels onto the tracer RNA in the coming months. So 488 and 647, functionally very similar to the existing product, just new colors for you to choose from. And so I think between the GFP, Cas9, and Cas12a, as well as these fluorescently labeled Cas9 guides, you have a lot of choice about how you label your RNP complex and how you can enrich for transfected cells. And all of that basically just amounts to getting higher levels of editing one way or another. And so those proteins and these new tracer RNAs should be available on the website in the next few months. Of course, if you have an immediate need, always feel free to reach out to me and we can arrange early access for those. The next section is really focused on gene knock-in and homology-directed repair. We've seen more and more of our customers moving in this direction because they want to be able to explicitly control what the mutant genome looks like. As I mentioned before, this is a harder experiment to do just because you're relying on a process that doesn't occur as often, and you need a donor template. But I think the benefits of it are what's driving people in that direction. So just by way of recap, a quick refresher, remember your nuclease comes in, introduces double-stranded break. And if you want to have explicit control over what the mutant genome looks like, you're going to want to take advantage of this HDR pathway. And so what we're going to talk about a little bit today is research on options you have for donor templates, as well as how to design these. So I'm going to start with the design side of things. This is a tool that is already on the website. It has been for a few months, but I think it is significant enough of a development to warrant repeating it here. When you're designing guide RNAs, obviously really, if you're using Cas9, all you're looking for is an NGG PAM site. Our online tools will help with other things like off-target analysis and predicting the on-target activity of that guide. But at the end of the day, designing a guide RNA is fairly straightforward. When you want to do a knock-in experiment, you need to design your guide RNA and donor template in conjunction with one another, because their positions relative to one another are really important. There are also lots of design rules around how long should your homology arms be, and how close does the cut site to the donor template need to be, and all of this sort of stuff. We have documents if you want to do that by hand, but I would highly recommend you use our design tool on the website. If you're working in one of our supported genomes, you can see on the right here, you're able to simply type in the gene name or the genomic location, and you'll get this interactive map where you can drag and drop the cursors, specify what mutation you want, either at the nucleotide level or the amino acid level. And then you basically click design, and we'll propose a donor template guide RNA pair that introduces that mutation into that genome. So it takes into account many, many rules that we've developed over the years based on our research on how to do these designs, and just automates all of it for you. So that's the URL there, and you'll find it on our website, but I wanted to mention it again because it really simplifies this process. Now, if you use the design tool, it's going to have some automatic recommendations on what type of molecule to use as your donor template, based again on our experience testing different options. And so this is actually some fairly old data that we generated early on based on feedback from customers and the types of templates they were using. So the relevant thing in the bar chart here is that the y-axis represents the HDR rate. So that's the rate that we're getting our donor template into the genome. And we're just comparing a few different options for synthesis of those donor templates. And so these two options you see on the right, megamers and ultramers, are really what we recommend today. So ultramers are synthetic DNA oligo. They're single-stranded. We can make them up to 200 nucleotides. So if the mutation that you're trying to introduce can fit within that length constraint, this is absolutely the best option for you. If you go over 200 nucleotide, you end up bumping up to our megamer products. And these can go anywhere up to 2,000 nucleotide. So both of them are excellent options from a CRISPR perspective, but admittedly, megamers have a couple considerations to go with them. One is cost, and the other is really synthesis time. They're one of our most complicated products to manufacture. And so as a result, it takes us a long time, and they do cost a bit of money. And so for that reason, today, we've seen a lot of our customers try out things like linear double-stranded DNA, which would be our G blocks instead. They do have some drawbacks, and I'll get into those in a couple slides, but they are more cost-effective, and we can make them more quickly. So sometimes we see that exchange happen. We've been doing a lot of research to try and identify better options, though. So I'll talk about that in a couple slides, because I think we have a great solution for that problem. I did want to start, though, just with a reminder on sort of recent developments for those smaller donor templates. The ultramers, those single-stranded DNA oligos, we recently launched a version of those that come with chemical modifications on the end of them. And the goal of this really was to improve the HDR rates. Again, that's what you see on the y-axis. And the way that this works is basically protecting the donor template from being degraded while it's in the cell. The longer you can stabilize your donor template, the higher your HDR rate, essentially. And so we sell 3 versions of these. The unmodified version is just a single-stranded DNA oligo, really not a lot different from a standard primer. We sell a version with a pair of phosphorothioate bonds on the ends. This is a really easy to incorporate chemical modification that also gives the oligo some exonuclease resistance. And so it helps stabilize it. And you'll find these in all kinds of IDT products, from our guide RNAs to our NGS library adapters, very widely used modification. And then the third option combines those phosphorothioates with the ALTAR modification, which is a proprietary blocking group we've developed. But the intention is to block exonucleases. And so obviously, based on the chart, that ALTAR version is your best bet. In terms of pricing, they all cost exactly the same. So we would highly recommend using this ALTAR version, unless you had some reason to use one of these. But this is clearly your best option for those small insertions. Now, as you get into the longer products or the longer insertions, I mentioned megamers has been our recommendation for a long time. Part of the reason for that is what you're seeing on the slide here. So I'll just walk through this a little bit. These figures are showing the outcomes of doing a CRISPR knock-in experiment when you use a linear double-stranded DNA as your donor format. So the desired outcome is really what you're seeing here on the sort of mid-left part of the figure. What happens in this case is you get a double-stranded break in the genome, and then your linear donor template is incorporated at the cut site via homology-directed repair. So the key thing here is that you end up with one copy of each homology arm plus whatever sequence you've inserted. So this is what you're aiming for. There is another possible repair pathway, and that relies on non-homologous end joining. And the problem with this is, implied by the name, it does not rely on homology between the donor template and the genome. What you have happen is just a blunt ligation of the double-stranded end of your donor onto the double-stranded end of the genome. And this results in a duplication of your homology arms in the mutant sequence. So if this is, in a protein coding region, obviously this is not good or useful for your experiments. The other problem with linear double-stranded DNA is that they can fairly easily incorporate by non-homologous end joining at off-target locations in the genome. So if your guide RNA happens to cut somewhere else in the genome, a single-stranded donor is not going to incorporate there, but a linear double-stranded one might by a non-homologous end joining. So if your CRISPR work has any ambition of moving into clinical applications or things like that, this type of outcome is probably not something you want to be seeing. And I'll show you on the next slide, this particular outcome here, the duplication of homology arms, is something that can actually occur quite readily. And so this is one of the main reasons we have not recommended linear double-stranded templates for some time. But our research group has been working on solutions to that. So let me walk you through the figure here. There are some experimental details on the right that you can read if you're interested, but I think that the interesting story is in the bar chart here. And so what we were trying to do here is knock in different sizes of inserts. You can see those along the bottom. And the y-axis represents the percentage of insertions that we're detecting. So anything between the bar and 100% means that some other mutation occurred or it was unedited. So the bar chart represents copies of the genome where we saw integration of the donor template in one form or another. The solid portion is the correct insertion, where we only have one copy of our homology arms. And this hashed section at the bottom is when we saw a duplication of the homology arms or a blunt end ligation event that did not rely on HDR. And so these are mutations that we really do not want to be seeing happening because they're not going to be useful for our downstream work. And so what you can see here is about 10% of the population, a little bit less at the larger size, but about 10% of the population is being mutated in that way. And so what we've been doing is trying to develop new chemical modification patterns that we can attach to the ends of double-stranded DNA donors that block that blunt ligation process. And so ALTAR1 and ALTAR4 are two potential chemical structures that we're studying at the moment. And you can see both of them do a really nice job of reducing those blunt end integrations. They also have the benefit of slightly improving the total editing rate. And this is probably because they help stabilize the donor templates as well. And this effect becomes even more pronounced at the larger insert sizes. So this is a really promising area of research for us. I think it presents a great solution to how we can make larger donor templates at a reasonable cost and with an affordable or with a reasonable turnaround time. without needing to resort to single-stranded DNA. Currently, we are still developing these, and I would anticipate them to be commercially available towards the end of this year. But again, if you have interest in things like this, let me know, and we can talk about providing early access to such products. On the HDR front, we've talked a lot about donor templates. There are lots of options for that depending on the insert size and things like that. But there are other things you can do to improve HDR rates as well. One that we've been recommending for some time has been to incubate your cells with our HDR enhancer molecule after you've edited them. So you deliver your Cas9, your guide RNAs, and your donor template, and then you incubate them in this molecule in the cell culture media for 24, 48 hours or so. And the way that this molecule works is just by inhibiting non-homologous end joining. So if you inhibit that repair pathway, cells tend to take advantage of HDR more. And you can see that when you compare the gray control bar to the blue bar, we get this nice improvement in HDR rates. So more cells are getting the donor template integrated in their genomes. There are a couple issues with this, one of which is that it is somewhat toxic to cell lines. Some cells are bothered by it, some are not. A big part of that is the relatively high dosing that we have to use with this. And so we've been working on developing a new version of this, which is a different compound that works through a very similar mechanism and can be delivered at a much lower dose. So it has two main advantages there. One, the lower dose means less DMSO, which makes it less toxic. And it appears that this new molecule actually works better than the first one. So it's a win-win on both fronts there. I think anyone who has not yet used version 1 should give it a try. We haven't yet launched version 2, and I hope we will in the next month or two. But if you're already using version 1, this new product should be a direct substitution for that, and you should see even better results as a result of making that change. Okay, so that was a little bit on HDR. I think HDR is really a huge area of focus for our research team. The other part of it that I'm going to talk about now is the result of our research efforts looking into off-target editing. And so over the years, our research team has done a lot of work on that front and developed many tools to help them do that. And some of those are now being commercialized and made available to our customers. And so the one that I want to talk about today is AmpSeq. AmpSeq is an amplicon sequencing technology that you would use with Illumina sequencers and can be used for many things, but we've noticed that a good majority of our AmpSeq customers are actually using this for characterizing CRISPR-based edits. So the main use that we envision will be people who care about off-target editing. So it's a robust way of measuring editing at many sites in the genome at the same time. But it can also be used as a high throughput tool for measuring on-target editing at multiple sites at the same time. So I'm going to talk through that a little bit and how it all works. So the basic experimental workflow here starts with identifying the genomic locations that you're interested in assessing. So usually, like I said, this is going to be your on-target site and any number of possible off-target sites, plus any other regions that you just happen to want to sequence at the same time. So there's a number of methods you can use for identifying off-target sites, from in silico predictive algorithms to GuideSeq to other methods. But the point is, to use AmpSeq, you would have to first identify all of those sites. Once you have those genomic locations, we'll design the AmpSeq panel for you, and I'll show you what that looks like on the next slide. Basically, the protocol is 2 rounds of PCR in the lab, and you put it on an Illumina sequencer to generate your data. And then I'll show you a little bit of what our analysis pipeline looks like. This is something that has not yet launched, but will be available probably later next month. So AmpSeq as a product exists today, but the analysis pipeline is coming next month. So if you're not familiar with AmpSeq, you may already be familiar with amplicon sequencing as a technology. If you know how amplicon sequencing works, you pretty much understand AmpSeq already. But just to briefly explain it, it's essentially 2 rounds of PCR where the final product is a ready-to-sequence library for an Illumina sequencer. So this first round of PCR is something that we designed specifically based on the genomic coordinates you provide. And so typically each pair of primers is going to flank perhaps the on-target editing site and any other locations like off-target sites that you want to study. This is a multiplexed PCR. So if you want to look at 50 targets in one reaction, this is going to be a 50-plex reaction. All happens in one tube, and we design the primers to be compatible with one another. And that's sort of the main advantage of using our design tool. AmpSeq is capable of supporting thousands of amplicons in a single reaction, but our CRISPR users are typically using no more than 200 sites at a time. So that first round of PCR generates a partial sequencing adapter. The second round of PCR is the one that adds your sample barcodes. And so you need to have a unique set of barcodes for every sample you want to multiplex and sequence together on an Illumina sequencer. And so once you've done that, you throw that on the sequencer, you generate your data, now you have to analyze it. And this actually has, our conversations with our customers has turned out to be a substantial bottleneck for many of them. I have this figure here, which is kind of intentionally meant to be overwhelming. The point is that there are a lot of tools involved in NGS analysis, right? A few of them are italicized here that you might use for cleaning up your read data, aligning it to a reference genome, and then calling those reads based on the mutations that are present in them. Setting up these types of pipelines typically requires some command line experience. You probably want to have a bit of a computer science or bioinformatics background to really understand what you're doing. So it's not trivial analyzing this sort of data. So what we've done is develop this pipeline in-house that we actually initially developed for internal use and have now sort of polished it up for our customers to use. We'll be rolling it out in a cloud-based platform. So really all that you'll have to do is upload your NGS data, tell us a little bit about your guide RNA sequence, and hit analyze. So all of this will be encompassed in that tool. Now, just to give you an example of this, this is a figure that's actually in a publication we put out back in 2018 describing our high fidelity Cas9. And as you can imagine, when we're talking about high fidelity enzymes, we're spending a lot of time talking about off-target editing. And so we actually used AmpSeq in this pipeline to generate all of the data you see in this publication. And this is just one figure to kind of show you the sort of analysis that can be done. What you're looking here are the on-target site in orange and a bunch of off-target sites in blue, and the rate of editing that we detected at each of them using AmpSeq. And so you can see that in the worst case scenario where you're letting the cells express Cas9, about 3/4 of the time, editing was at an off-target location. And by moving towards RNPs and using the high fidelity Cas9, we can almost eliminate that. And so this is just an example of what AmpSeq and the analysis tool can do. I think the other thing that is really important to remember about this is to generate the data required to make a figure like this by individual PCRs would have taken our R&D group probably months. Right? They estimated about 1,500 individual PCRs to generate this data. They were able to generate this using one AmpSeq run, which only PCR sequencing and analysis took a couple days. So it can really speed up the whole process for you. I did take a little screenshot of what the analysis tool looks like here, with a little caveat that this is a prototype version, so things might look slightly different by next month. But the idea is that once you've uploaded your data, run your analysis, you're going to be able to see results that look a bit like this. So really nice high quality figures about the amount of editing, the type of editing, where edits are occurring. All of these figures are exportable for publication or regulatory filing or whatever it is you need them for. And of course, you can export all of these values in an Excel file for further analysis. So I think this is going to be a really great way for people to measure editing when they need either to process lots of samples or look at multiple targets at the same time, or both. So that is coming next month. But again, as always, if you need access sooner, let me know and we can arrange early access. So with that, I'm just going to wrap up there and thank you all for attending. If you have questions for us, I would absolutely encourage you to start on our website. We have lots of great decoded articles and educational resources for you to learn more. The CRISPR email here comes to me as well as a whole team of technical support folks. So that's your best route if you need sort of general CRISPR support. You'll get the fastest response through that channel. And then if you have questions about today's presentation or just want to say hi, this is my email here. I'm always happy to chat with people who are interested.
With that, Marshall, are there any questions at this time?
Yes, we have a hand raised. Elizabeth Simpson, I'm going to unmute you from my end. So if you're unmuted on yours, you should be able to talk now.
Hi, Adam, excellent talk as usual. I had two questions. One is, what I do is using CRISPR in single cell embryos in mouse to generate mice. And I'm wondering how well the modification optimization that you do in cells like HEX and other things for guides and donors translates to the mouse embryo.
Yeah, that's a good question. I'm going to answer it a little bit more generally than just mice and sort of speak broadly to primary cells and other systems beyond what we work with. Because you're absolutely right that IDT's in-house research team focuses on easy to use immortalized cell lines like HEC293s and jerkats. Generally, what we find as you move into more, let's call them difficult systems like primary cells in mice, those modifications actually have an even greater benefit compared to unmodified guides, unmodified donors. The effects of nucleases seem to be more pronounced in those systems. So it's actually, I would say, even more of a reason to consider chemically modified reagents. The other question I often get in this sort of vein is, do we see toxicity as a result of these? The answer is generally no. I don't hear reports of that being a problem. Every system is new, but I will say that in mice, chemically modified guides and donors haven't appeared to be toxic. The only exception there might be megamers because they can be fairly long single-stranded DNA, and most cells respond with a somewhat toxic response to that. So dosing of those can be important, but generally I would say chemical modifications are preferred in all systems.
Okay, great. And my second question is, do you think you would be in the future making available the prime editor protein?
That's a great question. The short answer there is I would love to. Our research team has actually been working on that quite a bit. Obviously, selling the long guide RNAs and specifically mentioning prime editing guide RNAs kind of implies that. We've seen initial promise with that. I think before we start, beta testing and eventually maybe commercializing it, we want to be able to replicate that a few times. We're also interested in looking at more sites in the genome and different lengths of insertions because the initial work we've done has only focused on, I think, a one codon insertion. That showed promise, absolutely. So more to come on that, but I certainly hope we can in the near future.
Thank you very much.
You're welcome.
Great, thank you. Marshall, are there any other questions at this time?
At this time, oh yes, one just came in fact. And the question is, when the fluorescently labeled, when will the fluorescently labeled Cas9 be available?
That is another great question. I think, in a perfect COVID-free world, we would have had that available several months ago. But in the spirit of full transparency, we've been very busy accommodating a lot of manufacturing requests for COVID diagnostic reagents, which has delayed a lot of our product launches, unfortunately. I will say that product is essentially finalized and ready to go. We just need to make the bulk protein and put it in tubes. So I would anticipate that in the next 1 to 3 months at most. But like anything, if you have an immediate need for that, we can typically arrange for samples to come out of our research department. And that would at least hold you over in the meantime. So feel free to email me directly if you're interested in that.
Great, thank you, Adam. Any other questions, Marshall?
Presently, no more.
Great, that gives me a chance to ask a couple questions. So are there any specific chemical modifications that people should use when they're ordering the custom guide RNA?
Yeah, this is something that we've studied a little bit. It's always hard to provide specific recommendations on new CRISPR systems because they're all slightly different from one another. Generally for the longer guides, we've been recommending a very similar modification pattern to what you'd find on our Cas9 guide RNAs. So that's basically 3 phosphorothioates and 2 primomethyls on either end. And I can follow up by email on the specifics of that. We do know that occasionally that modification pattern isn't the optimal one. So I would always encourage people to take a quick look in the literature and maybe consider ordering 2 or 3 different variations on that pattern to see what's most appropriate for their system.
Great, thank you. And then the analysis tools that you were talking about, is there a way for people to use those if they're not using AmpSeq to generate their NGS libraries?
Yeah. So, the ideal solution that we're proposing with AmpSeq is that people use AmpSeq to generate their library and then take that data directly into the analysis tool. But the reality is the analysis tool will take data from any amplicon sequencing approach. So if you're using other tools like AmpliSeq or just generating the amplicons using regular PCR primers, that should be acceptable. We can sell access to that analysis tool on its own. So I would say get in touch with us. We can give you a few free analysis credits so that you can make sure the tool works well with however you've generated your data. And then we can go from there. But the short answer is yes.
Great. And then, so if someone's using an sgRNA instead of the 2-part guide RNA, can they still get those fluorescently tagged?
Yeah, so we've been adding fluorescent tags onto single guide RNAs sort of as special requests for people. Generally, the best recommendation that I've had before has been to use the 2-part system because that's sort of what we've optimized. And if you're using sgRNAs, I would say wait for the fluorescent Cas9s. We can, though, add fluorophores onto sgRNAs. The main challenge is that because it's a long molecule, and when you add a fluorophore on, you need to purify it, we tend not to get great yields. And so you get a pretty low amount of guide RNA for a fairly high price. And so that's really going to be the main advantage of the fluorescent Cas9. It's going to be just a much easier solution to that.
Great. And you're the right person to reach out to for working through stuff like this, right? Or at least you'll be able to direct them to the right person?
Yeah, absolutely. People can email me directly or just email CRISPR at IDT-DNA, and I receive those emails as well.
Great. And then about the HDR donor template, is there a specific length that the homology arm should be?
Yeah, we've done some research on this, and I think if you look in the literature, you'll find a lot of different answers, unfortunately. In our experience, the smaller ones, we typically use about 40 nucleotide on either side. And as the insert size gets longer, we tend to increase to between 1 and 200, depending on whether you use single-stranded or double-stranded DNA. The easy part, though, is if you use our HDR design tool, it will automatically do that for you. So you don't have to think too much about it if you go that route.
Great. And these products that haven't been launched yet, is there a way that people can try these out in advance?
Yeah, we're absolutely open to either having people beta test our products before they launch in exchange for some feedback on how they work. Or if we're already really close to launch and know how it's all working, we can usually provide early access. So the best path forward for that is to email myself directly, and we can just have a discussion to make sure it's suitable for your needs and set something up from there.
Excellent. Thank you, Adam.
No problem.
And Marshall, if there's no other questions at this time, we can actually close a bit early.
Yeah, no other questions presently, so.
Great. So in that case, thank you very much, everyone, for joining us today.